First off, let's make an overall view of the elements of the female reproductive system. It's certainly going to begin with the business end of the female reproductive system, and that's where oogenesis is occurring at the level of the ovaries. But then the ovulated oocyte will pass through a discontinuous duct system, including elements of the uterine or of the fallopian tubes, the uterus down through its neck, the cervix, vagina, and basically out through the vestibule associated with the external genitalia. The business end of the female reproductive tract are the ovaries, and basically oogenesis occurs in the area of the cortex, closest to the outer surface of the ovary. The medulla, on the other hand, basically contains the blood vasculature. When we actually look at the structure of the ovary, we can see in this illustration different stages of ovarian development. But we want to make the point that basically oogenesis begins in utero, and basically oogenesis not only begins in utero, but at birth, all of the oocytes are basically primary oocytes that are arrested in the prophase stage of meiosis I. And they're going to remain in prophase basically from the time of birth until basically follicular and oocyte development resumes at levels of puberty. So obviously the answer to the question of what's in the ovary at birth is basically all primary oocytes arrested in the prophase stage of meiosis I. So this is the point that the ovarian follicles are situated in the area of the cortex, and the oocytes themselves are surrounded by a secretory or follicular epithelium that is formed by either follicular or also known as granulosa cells. And basically, at birth, there are 400,000 primary follicles, but as we know, only a very small percentage of those 400,000 primary follicles will actually reach maturity and the adult and will be subsequently ovulated at the rate of 1 a month. The remaining follicles basically are degenerating progressively through atresia, and atresia can occur at any stage of follicular development, particularly in post-adolescent life. Secondary follicles, which basically begin their development at puberty, where the primary follicle completes meiosis I and begins to go through the stages of meiosis II, basically are characterized by the presence of a follicular cavity, a surrounding area of cumulus cells, a corona radiata, and basically an adjacent connective tissue area that contains the theca interna and external cell layers. The theca interna, basically outside the basement membrane that incorporates the oocyte, produces androgens that are then converted into estradiol by the granulosa cells that are basically inside the corresponding basement membrane. When the secondary follicle develops, what addition forms around that secondary follicle is a zona pellucida. The zona pellucida is a glycoprotein polysaccharide rich structure which must be penetrated by sperm and must be shed after fertilization occurs prior to implantation. A Graafian follicle is a definition of a secondary follicle that basically is about to be ovulated, and this follicle is large enough such that it extends to the entire cortex, and its structure is kind of illustrated in the micrograph on the next page. You can see basically in the center of the follicle the actual secondary oocyte. It's surrounded by its corona radiata cells. The area at the asterisk indicates the location of the zona pellucida. The granulosa cells you can see are outermost in this follicle, and they're separated by a basement membrane from the more external theca interna cells, both of which are obviously secretory in nature. And then finally, if we look over here, we can again see the stages across of how a primary follicle undergoes its development, the development of the secondary follicle, which will mature and form its Graafian stage. Recognize then what is actually ovulated. What is actually ovulated is just a secondary oocyte with its surrounding glycoprotein coat forming the zona pellucida, but recognize that that secondary oocyte is arrested in the metaphase stage of meiosis II, and only if fertilization occurs will that secondary oocyte complete meiosis II. So the answer to what is ovulated is a secondary oocyte arrested in metaphase. And the other question that's raised down at the bottom of this page is, where is the ovulated ovum immediately after ovulation? In other words, once it's extruded through the cortex of the ovary, it basically is now inside the peritoneal cavity, because there is a discontinuous duct system that does not actually directly link the ovary with the entrance of the fallopian tube. So in our next picture, we can see the proximity of the opening, the infundibulum, and its ostium, and its close proximity to the ovary, such that the ovum must pass briefly through the peritoneal cavity to reach the ostium, the opening of the uterine tube. So the infundibulum is the dilated distal portion of the tube. The portion of the tube that's longest and has the most clinical significance is the ampulla of the uterine tube. It's basically the most common site of fertilization, where the sperm penetrates the zona pellucida. It's also pathologically the most common site of an ectopic implantation into the wall of that uterine tube. Also shown is the point where the isthmus of the uterine tube enters the uterus, directly at the juncture between the body and the fundus of the uterus. And note that obviously the distal part of the uterus, its cervix, is inserted into the upper portion of the vagina. At the level of the cervical vaginal junction is what is known as the transverse cervical or cardinal ligament. This is largely connective tissue. It's traversed by the ureter, which lies perpendicular to the uterine arteries, but it's thought that this transverse cervical ligament helps prevent uterine prolapse, basically in postpartum females. The epithelium of the vagina is worthy of note. It's a stratified squamous epithelium that contains large components of prominent amounts of glycogen. Natural bacteria, basically in the vagina, break down that glycogen and convert it to lactic acid. It's that lactic acid that prevents proliferation of bacteria that have been introduced from the outside world into the lumen of the vagina. At ovulation, basically an increase in antral fluid pressure causes an increase in pressure on the wall of the follicle. Eventually the follicle ruptures and the ovum, along with its covering of the zona pellucida and its corona radiata, passes out of the ovary, and it must be fertilized within 24 hours or it's going to degenerate. The remaining portion of the Graafian follicle forms the corpus luteum, which we'll see is going to be converted to producing progesterone to help maintain basically the luminal epithelium of the uterus. So the follicular granulosa cells basically in that corpus luteum are the ones that actually begin to produce progesterone. If the ovum is not fertilized, basically the granulosa cells in the corpus luteum will stop producing and progesterone if they're not maintained by human chorionic gonadotropin, and it'll cause basically that corpus luteum to begin to degenerate about 7 days post-ovulation. And basically, if that ovum is fertilized, as we pointed out, the corpus luteum basically will increase in size for the next 3 months, and basically its progesterone output is then maintained by human chorionic gonadotropin that's produced by the syncytial trophoblast cells in the developing embryo. And following about the 40th day of pregnancy, the placenta takes over progesterone function such that the corpus luteum, basically, its functional role is decreasing through the balance of pregnancy.